Hello everyone, today's video is about DNA. Today we all are going to learn about DNA with an activity. So we extract DNA from a potato and then we are going to learn about DNA. So watch till the end because it's going to be very interesting. Everyone, please like, share and subscribe because it gives me motivation to make more interesting videos like this. So, first we are going to start with the activity of extracting DNA. For that, we need any soft vegetable which is mashed like potato, banana, strawberry, etc. So, here I have taken some potatoes. Then I have mashed it and took it in this. Then I have some water in which there is some diluted soap. So, this is a soap solution. Then we need an empty glass in which we can see the DNAs. Then we need some chilled alcohol which was kept in a freezer at very low temperature. And the last thing is we need a cloth. So let's begin our activity. So first we will add some diluted soap solution in the potato. Then we will mix it with a stick. So here we have mixed the mashed potatoes with the solution. Now this is ready. So now we will put the sticks aside. Then we will take this cloth and put it on the cup. It should be transparent. We cannot take strainer here because the potatoes can also go in. Now we are straining the solution in here. So now we have taken out all the water and the solution which can come out from this. Now we will give this also aside. At the end, then we have to take this some chilled alcohol, which is the chilled sanitizer. Then we have to slowly pour it on the side. We have to pour it that much that we can see a separate layer. As you can see, there is a separate layer. Now we have to wait for some time. Now, so after 2 to 3 minutes, we can see some part of the DNA. See, we can see some white lines formed on the above side of the orange color solution. So here we can see the DNA and now I am trying to take them on the sticks. Here you can see the DNA. Also, there are other small parts of the DNA inside. So here are the small DNAs that we can see. They are normally getting collected on the sides and some are also in the between. And inside you can see a small white line in this shape. So they are the collected groups of the DNAs. We can conclude that no living thing can be without DNA. Potato is a part of the plant and so we got some DNAs from it. And this is how we can get the DNAs from any food item. And if you want to get more DNAs of different things, then you can write in the comment. So what is DNA? Deoxyribonucleic acid is a polymer composed of two polynucleotide chains that coil around each other to form double helix. The polymer carries genetic information for the development, functioning, growth and reproduction of all organisms and many viruses. What does DNA do? DNA contains the instructions for making proteins, which are complex molecules that do most of the work in body. DNA is passed down from parents to offsprings, which is how genetically heredity occurs. DNA replication is the process by which DNA copies itself so that each new cell has its own complete genome. Where is DNA found? Most DNA is located in the cell nucleus, but a small amount of DNA can also be found in the mitochondria. How is DNA structured? DNA is made up of small units called nucleotides. There are four types of chemical bases in DNA: adenine A, thymine T, cytosine C, and guanine G. The nucleotides attach to each other to form chemical bond called base pairs which connect two DNA strands. DNA is shaped like a double-stranded helix held together by weak hydrogen bonds. Why to extract DNA? DNA extraction is a technique used to isolate DNA from cells or tissues. It is a fundamental procedure in molecular biology and life sciences. Purpose: genetic studies to understand genetic structure and function and to study the genetic causes of disease. Second, forensic science to analyze biological samples from crime scenes and identify suspects, victims, or establish relationships. And last but not the least, medical research and diagnostics to identify genetic disorders, study inherited diseases, and develop personalized medicines.